Okay, let's have a class, and this will be a review class before the exam. So because it's talk about the biochemistry, genetics, and also we talk about applied microbiology. So although it's an open book exam, but I think it's better we have some questions to let you practice and also remind you what type of the things we will test in the lab, okay? So in the class, sorry. So that's why we want to give you some of the sample questions you could be using. And you can bring these during the exam, that's fine. This is the first question is that what is FDA, FSIS, EPA? You don't have to worry about EPA because you can delete that one because we didn't mention about. So what is FSIS? What is FDA? So we talk about the full name, the FDA, which means Food and Drug Administration. And FSIS is Food Safety Inspection Services. So if you want to talk about what's a contribution in this area, then FSIS usually it's regulation of the poultry, meat, and processed egg. Is that right? Microbial safety, chemical safety, and FDA is all the other foods. Okay, so that's, you could look at is the slides, which is our we can look at this. We can look at this. Okay, wait a second. Oh, I forgot. I put in a wrong place. Sorry, I was messed up the. Okay, let me do this real quick. I don't want to mess it up because it's so glitchy yesterday evening. Let me download that first. Let me download this first, and then let me download that one. Okay, then. Let's go back to here. And bear with me a little bit. Exam 3. Practice 1. Bear with me a little bit. When I finish this, I'm going to add it. So we have this one right here. I'll add exam 2, practice 2, okay? So one by one. So then you have it. Okay. Yes, the same. Okay, so we talk about this practice question 1. Okay, second one. Which of the following processes generates the largest amount of the energy? So we have a fermentation, substrate level phosphorylation, anabolism, glycolysis, anaerobic respiration. First of all, we never talk about substrate level phosphorylation. Well, how many energy going to generate? It's just a different way to create ATP. Anabolism is using ATP, is not generating ATP. So you're going to be choosing only from fermentation, glycolysis, and anaerobic respiration. And we talk about the fermentation and the glycolysis. they are coupled. They are only 2 ATP generated. So, what is the best answer? It's anaerobic respiration. Now, if you say we forgot about where we could find it, then you could find the slides. The answer is right here at this lecture 3, the last slides. And you can clearly see here, the fermentation only 2 ATP generated. Anaerobic respiration is 36 or less ATP generated. And aerobic respiration is 38, but we don't have a choice here for aerobic respiration. So anaerobic respiration is a best choice, is a correct one, okay? Next one. The transfer of electrons and hydrogen by a dehydrogenase to an organic molecule as a terminal electron acceptor is called. The answer you can find is at this size. But like I said, this is very simple. Electron donor, electron acceptor. If the electron donor is an organic chemical, electron acceptor is also an organic chemical, this is called fermentation. That is the best definition. Now, if the electron donor is an organic chemical, and the electron acceptor is an inorganic chemical, this is called a respiration. And the respiration have 2, aerobic and anaerobic. That means if this final electron acceptor is oxygen, that's aerobic. Other than oxygen, that's anaerobic. That's very clear just right here. So always remember the best definition. And you can find it right here. Okay? And then what's the fermentation right here? So the answer here will be fermentation. Okay. We only going to have A, B, C, D. You're not going to have like F, D, F, G. We just. So the next question is the same. Which of the following is the best definition of fermentation? The oxidation of a carbohydrate with organic molecule 7 serving as a final electron acceptor. So the best answer is C right here. Okay. Next one. Which of the following is used as an electron carrier by living organisms? Well, that's all of the choices. We didn't mention the MADP+, but it's also is an electron carrier. So the answer is ABC. Okay. Which of the following can be used as an electron acceptor during anaerobic respiration? So go back to here. Electron donor is organic chemical. Electron acceptor is inorganic chemical, but other than oxygen. So that could be nitrate, could be sulfate, could be carbon dioxide. So all of the choices. And if you want to look at the slides, we could look for is right here. You can see on here there is an anaerobic electron acceptor. This is the aerobic electron acceptor. Obviously is oxygen, and anaerobic is have so many. Okay, so examples, and these are examples of the microorganism. So you can simply find that. Question number 7. The hypothesis that protomotive force driven ATP synthesis is called. This is simple. We don't need to know the process. We only need to know the name. So chemosmosis. Very importantly, when you do the exam questions, you're always going to look at the material, but also do not forget about the title. Sometimes is important, and that title could be just the answer of some of the questions, which make your life much easier. And this whole series is called chemosmosis. We mentioned it's created by Peter Mitchell in 1932. One of the theory talk about the proton motive force related to aerobic respiration. So you can see is the electron carrier, electron transfer during the electron transport chain. For bacteria is in the bacterial cell membrane system. Then coupled with the proton extracted out of the cell membrane system, then accumulated become a potential energy, which is called a proton motive force. And the potential energy could become a kinetic energy after they activate ATP synthesis, generate ATP. Then the proton finish their job, they're going to be transferred back to the cell membrane, inside of the cell membrane, and coupled with the rotation of the flagella. So that's called chemosmosis. But here, you don't even need to know the name. You don't even need to know the process. You only need to know it's called chemosmosis. Question number 8. Organisms that form lactate, ethanol, and carbon dioxide as end products of glucose metabolism are called. That's called a heterofermenter. This is something we talk about right here. And we said very clearly regarding this. Fermentation basically has two. Homo and hetero. Homo, basically you can see, generate lactic acid. And this is lactic acid. You don't need to know the structure, I'll just write to you. Hetero alcohol. That's CH3, CH2. Yeah, that's alcohol. Pretty simple. Okay. CH2. Okay, that's alcohol. Hetero-generated alcohol. So the answer here is heterofermenter. Okay, aerobic respiration. How many ATP generated? How you do the calculation? This I'm going to emphasize one more time. So like we said, we talk about the glycolysis. We talk about so-called TCA cycle. But I know some of you are non-major or not learned biochemistry very systematically. Are we still going to do it? How are we going to do it? So glucose become pyruvate, then become acetyl-CoA, then go from the TCA cycle. Okay, we skip all the chemicals, assuming we don't even know. And this here, how many ATP we used? Minus 2 ATPs. Okay, how many ATP created here? 2 ATPs and 2 ATPs. But don't forget, we said the glycolysis, if it's part of the aerobic respiration chain, there is a NAD reduced to NADH2 right here. There is also a NAD reduced to NADH2 right here, that means this is 3 ATPs. This is 3 ATPs. Okay, so how many ATP right here? -2 add 3 plus 2 multiplied by 2 equals 8 ATPs. Okay, now go here. What we need to find? We need to find the place, there is NAD reduced to NADH2 and the FAD reduced to FADH2. So where are they? Right here, there is NAD reduced to NADH2. There's another NAD reduced to NADH2. There is a GDP becomes GTP. There is a FAD reduced to FADH2. There is a NAD reduced to NADH2. Okay, so how many ATPs right here? You see that's 3, that's 3, that's 1, that's 2, and that's 3. So how many right here? 3, add 3, add 1, add 2, add 3. So how many? 3, there's another one here. So 3 add 3, add 1, add 2, add 3. That's 3 on the top. 3, 3, 3, 1, 2, 3. So when you add up together, that's 15 ATPs. And remember, there is a 2 circle because one part of it here, another one is here. So there's a 2 circle here. So this multiplied by 2 is 30 ATPs. And then you add up together, equals 38 ATPs. Okay, that's a very simple calculation. When we do this question, you write the brief procedure, what it looks like, and then we give you the points. Okay, so you don't have to write down all the chemicals. This one is just make fun for all of us. Just want to let you to thinking about question 10. If a yeast metabolizes 1 molecule of glucose to ethanol alcohol in the absence of oxygen, how many net molecules of ATP would be obtained? Well, it doesn't matter how long. A yeast metabolizes 1 molecule of glucose, become ethanol, become alcohol. That is fermentation. Regardless how complicated it is, fermentation, how many molecules of ATP will be obtained? Only 2. So the answer is 2. Okay, this just let you practice. We won't give you A, B, C, D, E, F, G, H, I, 2K. We only give you the choices A, B, C, D in the real exam. Okay, just let you practice. Because I believe the practice gave you a little bit tough question. We'll let you well prepared. So the answer is 2. Okay, next one. How are anaerobic respiration and aerobic respiration similar? Now we mentioned about that using electron transport idea. What's the similar? Both utilize inorganic molecule as terminal electron acceptor. Is that correct? Because if it's oxygen, we call it aerobic respiration. If it's others, we call it anaerobic respiration. That is always a very good answer. So the one, what I write on the blackboard at here, basically can do lots of the questions in this practice exam, and also could use to answer some questions in the real exam, because it's a very important concept and you should know. Okay, propionic bacteria makes probionic acids, acetic acid, and carbon dioxide in Swiss cheese. These products results from, well, this is easy, from fermentation. So let's say you forget. Do you remember Swiss cheese? Carbon dioxide have those holes and the propionic acids. Okay? So propionic bacteria organisms. Okay, let's go here. In aerobic conditions, it takes sugar to produce the same amount of the ATP when compared to the anaerobic conditions. Of course, less. Is that right? Aerobic respiration generates 38 ATPs. Anaerobic is somehow between 2 to 36. If you don't know, and you can even mark this possibly using the exam. And you can also mark these slides. That could be using the exam. Is that right? Because it's open book. You just don't have to remember, but then these two you can mark it. It will be used in the exam. Okay. So that's done for here. This set of the exam questions, let's let you practice. Basically is, we say, biochemistry. Okay, let's say the next set of the exam. I want to make sure I upload it because I go to the wrong section last time. It's a little bit glitch happened. So I'm going to make sure. Practice 2. Okay. Second set of the question in here, which is for the genetics. So let's go over these. Now, genetics kind of remind you, we have at our beginning of the third exam, slides slides 1 and 2. So third exam slides 1, which is we talk about genetics. These are basic information, and we also talk about replication. So let's look at the question. The 2 strands of DNA molecule R that is oriented in opposite direction. That's all running opposite direction. So what's the terminology for that? Anti-parallel. So remember we do this. What we say? Is that right? Anti-parallel. Opposite direction. We don't need this. Sorry. Consensus sequencing in the untranslated leader sequence of a prokaryotic mRNA, which serves as a ribosome binding sign, is called what type of sequence? SD sequence. Shai-Dagano sequence. This is the one we mentioned about in the slides. Let me show you where it is, because you can go ahead to review, when we go here, exam 2, and we mentioned you see it? The Shai-Dagano sequence. 16S RNA ribosome binding site. And you don't have to write the full name Shai-Dagano, just write the SD sequence is good. Okay. The amino acid tRNA synthesis categorize covalent addition to the tRNAs. Where are they? So where are they added? Just tRNAs. N-formyl methanol tRNA. The genetic formation of the DNA is divided into units called what? That's gene. Is that right? That's a gene. The genetic information of the DNA is called a gene. That's what's original definition of the gene comes. The basic difference between DNA and RNA resides in their sugar and pyrimidine base. RNA has ribosome and uracil, while DNA has deoxy and thymine. Is that correct? Yes. RNA has ribose. DNA has deoxyribose. RNA, instead of you have a thymine, you have a uracil, and DNA has thymine. So that's the answer is correct.
Sorry, what was the answer for 4?
Genes. Yeah. Which of the following is not a pyrimidine? So you have adenine, thymine, cytosine, and uracil. Which one is not a pyrimidine? Purine. Adenine is a purine, is that right? No, so that's not a pyrimidine. Cytosine, thymine, uracil is a pyrimidine. Which of the following is not a complementary base pair usually found in DNA molecules? A and U is not, because uracil is RNA. Is that correct? A and T and G and C. Which of the following is true about the structure of DNA? Well, we're going to have to see it. Purine and pyrimidine base are attached to the 1' carbon of the deoxyribose sugar. Purine and pyrimidine base are joined by phosphodiester bonds, and every term, every base pair is 0.34 nanometer. It's all of the choices. So, nanometer. This is the base, is that right? We added it here. It's 1', 2', 3', 4', 5', then you have a phosphate coming. Is that right? What's the difference? If you have O, that's RNA. If you don't have O, it's DNA, because it's called deoxyribose. So the base is at 1'. So this is all of the choices. Which of the following nitrogenase bases is usually found in RNA but not in DNA? Well, uracil, that's easy. When a DNA is replicated, the daughter molecules contain one strand of parental DNA and one strand of newly synthesized DNA. This is called semi-conservative replication. So, again, do not forget about when you do the review. Replication. Is that right? That's called a semi-conservative. Don't forget about it. Sometimes lots of people spending a huge time, but don't forget about the name called semi-conservative. Okay? And if you forget about puring and pyramiding, pyramiding, they have a bracket there. Okay? Just be familiar with when you do the exam. I know over the weekend, at least go over it. Don't just look during the exam time, because that's going to be cost you lots of the time there. Okay. The Y-shaped part of the DNA molecule, where the actual replication process occurs, is called a replication. We call it a replication fork. And this is what we have happened. Here is components of a replication fork. This is all the important ingredients we mention about every single function that all happen at the replication fork. So look at the title, sometimes it's important. During replication, the two strands of DNA molecule are unwound from one another by enzyme called, of course not B, of course not primerase. Primerase is synthesized primer. It's not a ligase. Ligase is connected Okazaki segments. It's called helicase. Now, this is a little bit tricky. If you forgot, then you're going to go back to see here to see the helicase. Okay? Then you find it here. Did you see helicase there? You didn't even find it. Because it's called, it is called DNA B. And then the helicase is right here. Can you see it? So sometimes when you have this question, you can even mark it there. Then the real exam have the same question. It's very easy to find the answer. So that's why I say the open book question is also a challenge for some of you. This is testing you where to find the answer. Because when you go future working the real life, the job, they will not give you an exam to ask you to do. But they always solve this real life problem. And what you do is you go online or you go textbook or library to find the answer. So that's why this is also a kind of test to you. Question 13. The coding sequence in the DNA of is normally continuously. That is not interrupted non-coding sequence. Now for this question is actually pretty easy. You have a prokaryotes. Okay, then you have a bee plants, you have a mammals, you have most insects. If you pick B, then you should pick C. If you pick C, you should pick D. It's a single choice, so the only answer is A. Because B, C, D, we gave them a full name called, a categorized name called eukaryotes. And their DNA is interrupted. There's an exon and an intron, so you have to cut the intron, the exon is connected. So the coding sequence in DNA is prokaryotes, is normally continuous. Okay, now we go here. The region at which the RNA polymerase combined is called. This is the one we're going to go here to find is transcription. So above the question is about the replication. Here we go transcription. And we said the transcription is very important, is this slide, which tells you a special region recognized by RNA polymerase. This is called promoter. So when you review, you can draw very simple. +1, -10, -35, this area, recognized by RNA polymerase. Then what's the name of here? Promoter. That's the answer. And if you want to know the sequence, you can just find it. So it's pretty easy. So promoter. The unexpressed regions of split genes are called. That's called introns. We won't test you this because we didn't mention, but usually it's a common knowledge. We have intron and exon, we connected them. But you don't have to worry about that. If we didn't mention, it won't be tested. Pyramidal box of E. coli, what's called a pyramidal box? -10. So pyramidal box is basically right here. That's called a pyramidal box. -10. So -10 equals we call pyramidal box. So the answer here, it's very simple, is centered approximately 10 BP upstream of the start site of transcription. So that's why we call it a -10, because it's 10 base pair upstream. Question number 17. The genetic code is said to be because more than one codon will specify a particular amino acid. What's the terminology for that? Not called redundancy, not called ambiguous, not called multiplicative and repetitious. It's degenerate. So if you forgot about this, you gotta go all the way to here. Did you notice that? 1 3-letter codon represents 1 amino acid. But 1 amino acid does not mean representing only 1 codon. And sometimes there is multiple. That's called degenerate because of the wobble effects. So the answer here is degenerate. Which of the following is not a termination codon? So we mentioned the termination codon, it should be very clear, is UAA, UGA, and UAG. And this initiate, we call it start codon, is always AUG, it represents methionine. So AUG. Molecular delivery amino acids to ribosome during translation. Which one? Transfer RNA. Is that right? Transfer RNA for the translation. The portion of the tRNA molecule that binds to the codon on the codon of the on the mRNA is called, well, that's called anticodon. If you doesn't know the answer, so you gotta go back to here. You can see very clear anticodon region. So that's why we give you 4 examples, because this is a typical question could be exam, and it's a very important function for transfer RNA. In ribosomes can attach to the mRNA and begin translation even though transcription has not been completed. Same thing. If you pick B, you're gonna pick C. If you pick C, you're gonna pick D. These are all called the eukaryotes, so the only choice is A, prokaryotes. Is that right? These practice questions are pretty nice. No matter what, you're going to get something. So, prokaryotes ribosome can attach to the mRNA and begin translation, even though transcription has not been completed. The reaction sequentially links adjacent amino acids to the growing peptide is called transpeptidation. That is happening right here. When you see this growing here, that's called a transpeptidation. Okay? Remember the 3 site, A site, amino acid binding site, P site, transpeptiding site, E site, and the exit site. We call it the EPA. It's a joke name for that, but you can remember. So here we call it a transpeptidation. Okay, the short answer. What are the biological functions of DNA polymerase 1 and 3 during the DNA replication? Since they are called polymerization, and there's an enzyme called polymerase, of course the function is polymerization. There's no doubt about it. Otherwise the enzyme will not come from there. Then what is the detail of DNA polymerase 1? What's a special for that one? Cut Okazaki segments. Is that right? Then what are special function of DNA polymerase 3 in addition to the polymerization? Is we said there is a fidelity problem right after DNA replication. And that rely on DNA polymerase 3 to do an important function. The first step we call proofreading. That's rely on DNA polymerase 3. So that's the answer. What is Okazaki segment say? This one you could go back to review. When we go here, I always say this is a very good slide. So Okazaki segments. What's Okazaki segments? A small piece of the DNA sequence synthesizing the lagging bands. And then they combine, they cut with RNA polymerase 1 and they combine with the ligase. Okay? Is that right? That's very, please look at this. That's very obvious what is Okazaki segments. Is that easy? The lagging strand is synthesized in short fragments called Okazaki segment. So it's not difficult. The answer is right there. And always don't forget, regardless of transcription, translation, and even replication, the direction is always from 5' to 3' only. Okay, this set is done. Okay, we're gonna go next set. Same thing. I will be adding the practice questions there before we go, because I want to make sure it's there. Because I don't want any of the glitch happen when I go home. Exam 3, like this. 3. So we added there. Exam practice 3. Let's look at the questions. What are they? Okay, this is something we mentioned, but somehow we didn't talk too much. So I will just tell you the answer, then you could use it. What is a coenzymes? That's a small molecule derived from the vitamins. Sulfur-made antibiotics that are functioned by inhibit the synthesis of vitamin by competing the normal substrate for the active site of enzyme involved in the vitamin synthesis. This is we already said. The transfer of electron hydrogen by a dehydrogenase to an organic molecule as a terminal electron acceptor is called fermentation. The purple sulfur and the green sulfur bacteria obtain energy through anaerobic respiration from reduce this to here. This is a little bit difficult question. If these in the exam, then usually it is hard to find. But where it is right here. The name of those sulfide is called disulfur-vibrio, disulfur-tamacule. It's a very interesting name. Disulfur, look at that. That means sulfite is reduced, and the morphology is vibrio. That's environmental bacteria. Which of the following is the best definition of fermentation? We mentioned already, repeated. The oxidation of a carbohydrate with organic molecules serving as final electron acceptor. This we talked about. Is that right? We just mentioned disulfur-vibrio is using H2S as its terminal electron acceptor. Which of the following reaction takes place outside of bacterial cell? So they remove out of the bacterial cell. Then you're going to go back to here. Dehydrogenase. What means dehydrogenase? Look at the word's name. Take the proton out, become proton multiphos. Take them out. So that gives you the answer. So you look at it here. Removal of electron and proton from organic molecules by dehydrogenase. Is that right? The picture exactly showed what the words meaning. This is easy. Oxidation of a carbohydrate, organic molecules serve as the final electron acceptor. And I intentionally to have these questions because I want to have the concept with electron acceptor and the donor. That is new. Some of you don't understand it very well, but when we do it, we know. The conversion phosphoenolpyruvate to pyruvic acids, what is called, involves conversion ATP to ADP.